So, good morning. My name is Victoria Gillam, and I'm a rising sophomore at Howard University, studying clinical laboratory science with a minor in biology and Spanish. And this summer, I was working at Boston College's RU program in Dr. M.J. Gubble's lab under Dr. Daniel Tago, working with the obligate intercellular parasite Toxoplasma gondii. And more specifically, we're working on the secretion of a protein within Toxoplasma gondii called DDS1, but formerly known as TGG-T1 209600. So in my talk, I'm going to tell you what is Toxoplasma gondii, how DDS1 impacts toxo, our methods, results, and future results for future research. So, like every parasite, toxo has various hosts that it infects, and even though it is not on this picture, I want to highlight one of the ways it can infect a person is through immunocompromised individuals because they cannot fight the parasite. But in this image itself, there are various pathways that toxo can infect its host. The most well-known example is through a cat's litter box, and this is most well-known because pregnant women either have to test their cats or they can't take care of their cat's litter box because if infected, it can negatively impact the fetus. Another example is if prey ingests the oocytes of toxo and then it is ingested by the predator. That's another way to infect the host. And another way, if livestock also ingest the oocysts and then their meat either isn't cooked properly or consumed rawly by a person, then that person can also be infected. So what I'm specifically looking at is the lytic cycle of toxo. And the lytic cycle is a cycle that actually gets you sick. And within this little cycle, toxo is in its first stage when the parasite is a tachyocyte. And a tachyocyte is a crescent or oval-shaped form of a parasite where it invades the host cell and replicates until the host cell is kind of swollen and then lyses out and re-invades. So with this, we were looking at tachyocytes itself because it rapidly multiplies. So these two images kind of depict the necessary components of toxo. The first image is what I'm specifically looking at, which is microneum secretion. So microneum secretion allows toxo to leave or egress from the host cell and glide into a new host cell. So we were looking at this because a protein called DOC2 blocks this microneum secretion, and below is a graph that dictates when and where microneum secretion and other organelles are used within the replication process. So you can see how in the red is microneum secretion, so you can see it spikes during egress or leaving the host cell, and then kind of tapers off when it's free moving, and then spikes again when it's reinvading. And this is done because calcium, which is this cream line, is a catalyst for the microneum secretion. So it mimics the microneum band, as you can see between these two, whereas it doesn't really depend on the other two, as Rothschild bulbs and dense granules allow the parasite to re-enter the host cell and then start its replication process. So, as I said before, we were looking at microneum secretion because DOC2 blocks microneum secretion. And DOC2 is a calcium sensing protein that binds and triggers the release of micronemes. So with DDS1, aka DOC2 dependent secreted protein, it we saw that within these two graphs, there is no secretion, but DDS1, well here's DDS1, the 40 degrees, and there's no secretion. And it's the only protein that has no secretion, whereas other proteins, they either do have secretion or they don't secrete at all. So my goal is to figure out why and the impact of that. So our aims was to tag the gene DDS1 with MYC and a YFP expressing protein to localize its location within the parasite to future organelles and see that process, and determine its localization by expressing the gene in temperature-sensitive conditions, such as the last slide, whereas in 35 degrees the microneme secretion was available versus 40 there was no microneme secretion. And then lastly, we want to validate its increased release of microneme secretion with immunofluorescence assays. So the first method in validating and trying to figure out the purpose of DDS1 is a PCR, and we did this by amplifying the promoter gene using Rh genomic DNA. And once we confirmed that our PCR was working, we then moved on and tested that PCR product in a digest, which means that we used 2 restriction enzymes and cut out exactly what we wanted, and then used a ligation, which means we took that specific piece that we cut out and then reinserted it into the vector that we wanted to make a full circle. So we did that with the 2 expressing proteins, the YFP and the MYC. So here's a picture of the PCR that we did, and we confirmed it, and then moving on into the digest and ligation. And after you reconfirm the ligation, you can then do a transformation, which is then putting your vector or plasmid into a bacteria culture. And these are results of that bacterial culture itself. So there's about 24 conformations that we use just to make sure. So the first 12 is the MYC protein that we use to tag it, and the double bands we sent to sequencing, but then we had to retest the single bands because that means it wasn't expressed properly. And along with the YFP, we sent the double bands to sequencing. And here's an image of this sequencing, and we checked the alignment because we know the specific genome of YFP and MYC, so we tested it against each other to see if it matches up. So if the alignment was correct, we could move on. So the next method is the transfection process, which takes the longest. So you start with your parasite, which is toxo, and you insert the plasmid that you want, and then you do a gene pulsar, which means that you put that specific plasmid into electrocution process, and that allows electroporation, which temporarily kind of opens up the cell membrane that allows the plasmid to kind of enter in temporarily, or not temporarily, but enter in before the cell membrane closes back up. And then you do a cell culture under select drug conditions. So that means that with certain bacteria, if they do not have that drug, it'll die. So that kind of separates the process. So you know for certain that the bacteria or the parasites that you're left with definitely have the drug and they're resistant to that. So these are the parasites you can test under immunofluorescence assays. So this is the first immunofluorescence assay I did with DDS1 and the YFP protein expressing. So this is the YFP tag, which is green, and VP1, which stands for DDS1, and DAPI, which is nuclei. And here's a merger of all three, and we want colocalization to figure out which organelle it's going to be in within the parasite, and that's used to figure out its function. And the second picture, which is DHFR T7S4 TYDDS, is used for a previous cell line that was made when I wasn't here because, as I said, transfection does take days and you have to make sure the parasites lyse out completely and have the selected drug markers that you can use. So then I started looking at another cell line that was previously made. So first stain is TY, which is standing for a basal complex, and then INC3, which is the outer cortex, and DAPI, which is nuclei. And here's a merge of all three within the parasite. And we're not sure whether it's in the mitochondrion or the Golgi. And the last similar previous parasite line that was already made before I got here, we use another staining called NACT, which is a Golgi marker, and here's a DDS1 and DAPI. However, the staining didn't colocalize in the Golgi itself, so that's the question of what other antibodies should we use to get that colocalization. So in conclusion, the IFA shows different colocalization, and this may be because of different plasmids and the different antibodies we use. So in order to figure out which antibodies actually fit, we have to use more than one. And then the next is IFA of GDS1 and YFP also shows different localization within VP1 that was shown in the previous. So future, we want to deplete the protein levels and determine its effect on the parasite because, like I said earlier, microneedle secretion, once it's blocked, the DS1 is increased, so we still want to answer that question. And then transfect the 3 times MIC into DS-DOC2, which we didn't do because the MIC, the drug selection wasn't working and the parasite wasn't lysing out correctly, so then we just focused on YFP. So we do want to go back and do that. So as an acknowledgement, I would like to thank Dr. Birch for allowing me to participate in this RAU program, and Parami and Dr. MJ Gubbles-Lab, and along with Dr. Tago for teaching me all of these methods that I've never done before, and the other RAU students that I've met and giving me new perspectives. And that is the end of my presentation.
So I have a question about your imaging of DDS1. So you said that the construct that you were using is YFP labeled, is that right?
Yes.
So when you're imaging, were you imaging using at all live imaging of just the YFP, and how did that relate with the immunofluorescence of the fixed?
So we didn't do any live imaging, it was just fixed imaging.
And is there a reason why you didn't try live?
So for about a month, our microscope was broken. So then when it began working again, I just focused on fixed imaging.
Other questions? If you would do a slide with some gel sequencing results, and yeah, right there. Did you know why those bands are not really clear, defined horizontal bands? They're kind of circular / kind of smeared.
Well, I think it's because the amount of time I let the gel run.
Okay.
I also had a question. So at the beginning, you were talking about this business where you had this ring, and then you were opening it and filling it. What exactly was the ring?
So this was the plasmid or vector itself, and we wanted to take the 2 restrictive enzymes from the digest, and once you cut them up, it opens the ring up itself, and then you can use a ligation to put that product into the ring and close it back up. So that's why it goes from these 2 blue and red, which is the 2 restrictive enzymes, opens and then enters the whole kind of process, and then it's just blue, red, and green.
Any other questions?
Yeah, Melissa.
Can you go back to the slide where you have the cells, the diagram?
Wait, which,
Like the one that the organelles are labeled to.
oh yeah, sure.
So can you explain exactly what a microneme is?
So here are micronemes, and we know that micronemes itself help the parasite egress or like leave. So I know it's a kind of cylindrical organelle that help the parasite leave itself and then glide into a new parasite. So it's kind of like a process that without it, it wouldn't be able to lyse into a new parasite.
But like, do they have any function within the cell?
Other than helping it leave, not that I know of, no. But I can totally look it up for you and come back to you after during the break, I can talk to you about it.
Thank you.
Can you go to the imaging of the cell with the labels?
This or,
With the microscope.
okay. So this.
What scale is this? Like how big is the image or the cell?
It's not big at all. I just cropped it to make it look closer than it actually is. But when you're looking at it in the microscope, it's not big.
But like how, do you know how many like microns or micrometers or?
I'm not sure, but if I were to answer, I would say 6, 7.
Okay, if there are no other questions, let's